Good day, viewers. Welcome to another biology presentation. So in this presentation, we are going to look at biology paper 2 for 2021. So in this presentation, we are going to answer question 1 up to question 3. So let's see our question 1. So here is our question 1. Our question 1 reads, Figure 1.1 and 1.2 show clearly fresh cut potato chips suspended in solution F and solution E at the beginning of an experiment and after 2 hours respectively. So we have our figure 1.1 here, and we also have our figure 1.2 right here. So in figure 1.1, we are looking at the beginning of the experiment where we have solution E and solution F, and we have the fresh potato chips which is cut, which is in solution E, and another one in solution F. Then after 2 hours, we have the same solution E and solution F, but the structure of the potato chips has changed. So let us look at question 1A1. Explain the difference in the appearance of the potato chips in solution E and solution F after 2 hours. So you can see at the beginning of the experiment, the potato chip in solution E, it was bigger, okay, and after 2 hours in solution E, you can see that the potato chip has become what? Smaller. Meaning that this potato chip in solution E after 2 hours, it has lost water by osmosis. That's why it has even become smaller. And then when you look at the solution, the potato chip in solution F, okay, after 2 hours, you can see it has become bigger and it has gained a lot of what? Water. So our answer here to explain the difference in appearance of potato chips in solution E and F after 2 hours, we can say that potato chip in solution F loses water by osmosis and becomes smaller, while potato chip in solution F gains water by osmosis and becomes what? Bigger. That is our answer. So I hope that you have read it's clear. So this is our answer. We can now move on to our second question. Okay, so our second question is here. Based on the appearance of the potato chips in solution E after 2 hours, what term could be used to describe solution E? So here they are asking us that when you look at the appearance of potato E, the way it's appearing right here, after 2 hours, what term can be used to describe solution E? So we have said this potato chip in solution E, it has become smaller and it has lost water by osmosis. So the term that can be used to describe solution E, it's hypertonic solution. Okay, it's hypertonic solution. So that is our answer there. Let us now move on to our question B1 right here. Our question B1 reads, what would be the state of cells in the potato chips in solution F after 2 hours? What would be the state of cells in potato chips in solution F after 2 hours? So after 2 hours, the state of cells in solution F would be that it's going to gain water, it's going to swell, it has become big as we have seen. So the state would be turgid. That's easy, the state. Okay, so our answer there, we have said the state is going to be what? Turgid. Okay, then B2, state the importance of the state of cells in B1 above to plants. So what is the importance of the turgid to plants? So turgid, we are saying this is a situation whereby a cell gains water, okay, becomes swell. So what is the importance of this state? The importance of turgid state is that it provides support to the plant. That is our answer. Okay, so we have said it provides support to plants. That is the answer. So let us now look at our last question here, which is question C. Explain the meaning of the term isotonic solution. Explain the meaning of the term isotonic solution. So when you talk of isotonic solution, we are looking at the solution whose concentration is equal to that of the concentration that is inside the what? The cell. So we can say that isotonic solution is the solution that has the same water potential, okay, as the cell inside the what? As the cytoplasm inside the cell. That is what isotonic solution is. So we have said isotonic solution has the same water potential as the cell cytoplasm. So we are done answering our question 1. Let us now move on to our second question. So this is our question 2 right here. So under our question 2, we have question A. Table 2 shows various food tests carried out by a learner. So this is table 2. So in this table 2, we have nutrients, test, observation, conclusion. So under nutrients, we have starch. The test, there is blank. Observation, color changes from brown to blue-black. Conclusion, starch is present. The second nutrient is blank there. The test is emulsion test. Observation, white precipitate. Conclusion, it's also blank. Under the same nutrients, we have proteins. The test is burette test. The observation is blank. The conclusion, protein is present. So our question 1 is here. Complete the table by filling in the spaces. So we are going to start with starch here, the nutrient starch. The test for starch is the iodine what? Test. So here in 1 we are going to have iodine test. Okay. Then we come to nutrient 2 where it's blank. The test is emulsion test. So emulsion test, we normally use it to test for fats or lipids. Then the observation, white precipitate. So just because of this observation, our conclusion is going to be that fats are present, okay, or lipids are present. The last part we have is under nutrients, we have protein. The test is burette test. The observation of proteins is that the color changes from blue to purple, okay. Then conclusion, protein is present. So that is how you can answer that question, okay. So we have said iodine test, fats or lipids, fats present, the color changes from blue to purple. So we have answered our question A1. Let us now look at our question A2. Which nutrient in the table would cause kwashiorkor if lacking in the human body? Which nutrient in the table would cause kwashiorkor if lacking in the human body? So the nutrient in this table that would cause kwashiorkor if lacking in the human body, it's what? Protein. Okay. So our answer here is protein. This is our answer. So I hope that you are following. Okay. So proteins. Then we come to our question A2. Name a nutrient not in the table which helps to prevent constipation. So the nutrient that is not in the table that helps to prevent constipation, it's roughage, or you can say dietary fibers. Okay, that is the answer. So let us now look at question B. Under question B, this is the question. State 2 deficient diseases in plants. So deficient diseases in plants that we have, they come as a result of plants lacking either phosphorus or nitrogen in the soil. So these deficiencies can either be where the leaves will be start turning yellow because there is no presence of chlorophyll. Again, we can see leaf flaking. Okay, so the 2 deficient diseases that we can have in plants is, we can say, leaf crosses and the leaf flaking. So under leaf crosses is just a situation whereby the leaves start appearing what? Yellowish because of the absence of chlorophyll. So we are done answering our question 2. Let us now move on to our last question, which is our question 3. So this is our question 3 right here. Our question 3 reads, Figure 3 shows a longitudinal section through a root tip. So this is figure 3. which is showing a longitudinal section through a root tip. So we have the vesicular bundles, we have the structure labeled G, H, I, and J. Our first question is identify the parts labeled G and J. So the part labeled G is called the root hair, okay, and the part labeled J is called the root cap. So these are the answers, okay. The part labeled G is the root hair, and the part labeled J is the root cap. So let us now move on to our question B. Our question B reads, which labeled part in figure 3 is responsible for generating new cells? So they want us to identify the labeled part in this figure which is responsible for generating new cells. So new cells are going to be generated at the region labeled what? I. That is where we have the meristemic what? Cells. So I hope you have heard what I've said. So new cells are going to be generated at the part labeled what? I, which is part I. We now come to our question C. Our question C reads, region H is responsible for cell differentiation. C1, apart from vesicular bundles, state two other types of cells differentiated by region H. So there are many cells that are differentiated by region H. So they just want us to mention two. So you can say sclerenchyma, also parenchyma, and the chorenchyma. So they just want two, okay. So if you mention N2, they will give you a mark, okay. So any of the two you can mention, but don't mention xylem and the phloem because they have said apart from the vesicular bundles. So here we have said we have sclerenchyma and parenchyma. We also have chorenchyma. So you can just mention any two. So we come to our question C2 right here. State two types of cells found in a vesicular bundle. So a vesicular bundle consists of the xylem and the phloem cells. Okay, so those are the answers. I hope it's clear. So we have answered our question. Let us now move on to question C3. Which named type of cells in C2 allows movement of substances upwards and downwards depending on the needs of the plant? So here in C2 we have said we have phloem and xylem. So which of the two allows movement of substances upwards and downwards depending on the needs of the what? Of the plant. So the cell that will allow movement of substances upwards and downwards depending on the needs of the plant is the phloem cell. So that is our answer. We now come to our question C. So our question C reads, so this is our question C. I hope you are able to see it nicely. Suggest one meristemic structure, not in the diagram, responsible for bringing about secondary growth in dicotyledonous plants. So one meristemic structure that is not in the diagram and is responsible for bringing about secondary growth in dicotyledonous plants is the cambium. Okay, that is our answer, cambium. So I hope it's clear. You have gotten what I've explained. So we have now come to the end of our session. Thank you so much for being so attentive. This has been your presenter, Mr. Mlenga. Bye-bye.